Hello, and welcome back for part 3 of our look at brain anatomy. And in this video, we're going to be focused really on kind of accessory structures. We've gone through the major areas and regions of the brain. Now we just got to kind of tie up some loose ends. And one of those loose ends, but yet extremely important, are the meninges. And the meninges serve a couple different uh, functions. One is to cover and protect the central nervous system. And so understand that the meninges that we're going to be discussing are not only surrounding the brain, but also the spinal cord. All right. They also protect the blood vessels and they enclose the venous sinuses that are traveling around the brain to, again, collect waste, debris, and remove that from within the cranial cavity, again, through venous blood supply or the veins. Um, the meninges is also where we find cerebral spinal fluid, which we're going to talk a little bit more about in a bit. All right, and it also forms walls or divisions within not only the skull but the brain as well. And what we will see is that the meninges are divided into or have three main layers to them: the dura mater, which is the most superficial layer. In other words, it's external. The arachnoid matter, which is in the middle, and the pia mater, which is the deepest of the layers, and in fact is directly covering and attached to the brain. And so we will spend the next 20 minutes or so not only learning about the meninges and cerebral spinal fluid, but a couple other little things as well. And so this is a really nice illustration here or diagram of those key terms and structures that you are going to be responsible to know. And I'm going to go to a highlighter here. And so what do you need to know as far as this model or this diagram here goes? Well, you need to be able to identify the dura mater, all right, right there. All right. You do not need to know the periosteal and meningeal layers of the dura mater, but you do need to know that the dura mater is the most superficial of the layers. It is the thickest and toughest of those layers. In fact, the dura mater is translated into meaning tough mother. Matter meaning mother, dura meaning durable or tough. And we're going to talk a little bit more about this coming up on a few slides. You also need to know the arachnoid. My thing is still acting up. I don't know why. You also need to know the arachnoid matter. Right? And the arachnoid matter, arachnoid meaning spider-like. Right? And so the arachnoid matter has what we define as being, I don't know why this is really just acting up today. The arachnoid matter containing the arachnoid space that you are seeing right here. Right? And this is really where it gets its name from, because the arachnoid space has these extensions that come off of the arachnoid matter, and they almost look like little spider legs. And again, this is kind of where we get that idea or that notion of um, the arachnoid. And I apologize for all the craziness that's happening with the highlighter today. You also need to be aware of and be able to identify the pia mater, right, which once again is directly attached to the brain, right there. Other things that you need to be aware of within this model. The superior sagittal sinus you need to be able to identify. All right. These things here called the arachnoid granulations. Right? Sometimes you'll see them referred to as the arachnoid villi, V-I-L-L-I. But that's these little things that are right here. And what happens is those arachnoid granulations collect debris from within the cerebral spinal fluid, right? And they diffuse into the sagittal sinus, which is really a vein. Right? And this is how we start to get rid of waste products from within the cerebral spinal fluid. I will also mention that it is within the subarachnoid space that cerebral spinal fluid is found. Right? And it is also where we have blood vessels, both arteries and veins. I don't know if I've mentioned this before, but um, the neural tissue itself, the gray matter and white matter, does not have any blood vessels within it. All of the vasculature to the brain is all external, and it's all kept within that subarachnoid space. All right, here's that term right there, subarachnoid space. All right. And then we have that fissure. Right, we have that fissure right here. And this is what we refer to as, and this is an extension of the Dorma matter, which is called the falx cerebri. That is right there. All right, so the longitudinal fissure would be the space, and the falx cerebri would be what is going down into that longitudinal fissure. And so we're going to actually look at each of these in a little bit more detail. Right, that you're going to see here. I'm going to try to not use my little writing pad since it's not cooperating with me today at all. As we mentioned, or as I've mentioned, the Dorma matter is the toughest of the three layers of meninges. Okay, you can actually see that Dorma matter right here. Right, this is where it's been removed, but this is it there. It's a milky white, very leathery, tough covering, and it's designed to kind of anchor the brain within the cranial cavity. Right? So in other words, your brain can't just kind of freely shake, rattle, and roll within the cranial cavity. It's suspended within that cavity to try to prevent damage from occurring. And you'll notice that the Dorma matter has three major divisions, right, or separations, what we call septa. Right? One of those, the falx cerebri, we've already talked about. That would be this area right here. Right? The next one is the falx cerebelli. Right? The falx cerebelli, which is this extension down here. So that would be this division right here. Right, so that's what's dividing really the lobes of the cerebellum, which is what you're looking at there. And then the tentorium cerebelli, which is this division coming in right here, and it actually extends left and right. And so that tentorium cerebelli would be separating left and then going this way as well. Right, so you have the falx cerebri, which is up here. Right, this would be the falx cerebri. You've got the falx cerebelli, which is this extension down here, and then the tentorium cerebelli, which is moving left and right in that area. The arachnoid matter. Right, the arachnoid matter, which again is this white line right here, right, contains the subarachnoid space, which we've already identified as being the area that has cerebral spinal fluid flowing in it. Right, those arachnoid granulations or arachnoid villi. Let's see if my pen's going to be cooperating here. I do apologize. I do like writing, and this is just not working. And so again, another term for the, yeah, this is not working, guys. Another term for the arachnoid granulation is the arachnoid villi. Right, so you will see both of those terms used interchangeably. Right, so don't be freaked out by that. Um, and again, you can see that there's the blood vessels that are within that subarachnoid space as well. And then the only other layer that's really here is the pia mater, right? Pia being delicate, right? And so this is the delicate layer, and that's because it is the thinnest of the three layers of the meninges. It is directly adhering to the brain. You can see it there. When you actually look at the brain, to separate the pia mater from the cerebral cortex is not easy. If you have very steady hands and a very um, pointy, sharp, high quality forceps, you could peel back the pia mater. But what I usually do in the lab is I have students take the cerebral cortex and squeeze it in their fingers, and the pia mater usually crinkles up, and you can see that in the light. You can actually see the crinkling of that in the light. And as I've mentioned, cerebral spinal fluid is what is flowing through that subarachnoid space. And think of it, the cerebral spinal fluid, as kind of it bathes the brain and allows the brain to float as well. And so as the brain is floating within this cerebral spinal fluid, it's providing protection. Right? It's providing protection. Um, and so it gives the brain buoyancy, it floats, it provides protection, and it also provides nutrients for the neural matter as well. And so, as we will find, uh, nutrients, glucose, hormones, um, other chemicals within the body have the ability to cross into what we call the blood-brain barrier and cross into the cerebral spinal fluid, but it's highly selective. And so we really, really, really try to protect what is in that cerebral spinal fluid at all times. And there's a reason for that. And part of that is because within blood, we have a lot of neurotransmitters that get released. Um, uh, Glutamic acid, glutamic acid, aspartic acid, glycine are three neurotransmitters that are usually found within the blood and interstitial fluid, but they are absent from cerebral spinal fluid. And part of that is because what glutamic acid, aspartic acid, and glycine do is stimulate neurons. So imagine if those three neurotransmitters were able to cross the blood-brain barrier and get into the cerebral spinal fluid, your brain would be misfiring all day long. Okay? And so this is one of the reasons why we have this thing called a blood-brain barrier. And that is to regulate really what is coming in and coming out. Um, and it is it's overall for the protection of the brain. Um, it is the cerebral spinal fluid when they do a spinal tap. That is what they are. That's what they're removing. They're removing that cerebral spinal fluid to usually analyze it for anything that might be unusual. Cerebral spinal fluid is highly sterile. And so any bacteria that might be in it, when we talk about bacterial meningitis or viral meningitis, meningitis is referring to the meninges. And so that's a bacterial or a viral infection specifically within the cerebral spinal fluid that is causing inflammation within the meninges, within the arachnoid matter specifically. Um, and just as a FYI, um, about 40% of the cerebral spinal fluid is produced within the subarachnoid space. 30% is produced by the ependymal cells lining the ventricles. We haven't talked about the ventricles yet. We will. And the other 30% is produced by the choroid plexus itself. And we're going to talk a little bit more about that coming up here in a few moments. All right. And so this is a nice diagram because it actually shows the flow of cerebral spinal fluid. Okay, again, here are the arachnoid villi. And again, the idea is we collect waste in here, and then that waste diffuses into the sagittal sinuses, or the sagittal sinus here, which is venous blood supply. And so we're getting rid of that waste material. All right. But when we look at the flow of that cerebral spinal fluid, we also have to take into account something called ventricles. And ventricles are spaces within the brain that allows for blood to get close to that neural tissue, but not come in contact with it. And so we've got 4 ventricles within the body. We have a lateral ventricle that you see right here. And there's 2 lateral ventricles. There's a lateral ventricle on the left in the left hemisphere. There's a lateral ventricle in the right hemisphere. So there's 2 lateral ventricles that you're seeing right here. There is the third ventricle that is right here. Right? And so the third ventricle is kind of completely surrounded by the thalamus, the epithalamus, and the hypothalamus. Right? Otherwise the diencephalon. And then the fourth ventricle is down here, that is posterior to the medulla oblongata. All right, but it's anterior to the cerebellum. And what's unique about these ventricles, right, besides being spaces where cerebral spinal fluid can acquire nutrients, it's also where we have something called the choroid plexus that is located. And the choroid plexus is it's a group of capillaries. It's a clustering of capillaries that hang within the ventricles located throughout the brain. And so if I come back to this image here, again, here's the lateral ventricle, the third ventricle, and the fourth ventricle. You see that red cluster right there, and right here, and right there? That is the choroid plexus. That's that network of capillaries. All right. And so you can see that within that ventricle, this capillary is going to come down. All right, and the natural tendency is for things to diffuse out of the capillaries. Well, to ensure that we've got regulation and control, kind of like a border security check, if you would, these choroid plexi are lined with ependymal cells. And so here's our ependymal cells that we talked about in a way earlier video. These are the ependymal cells. And the job of those ependymal cells is to filter anything that is coming out of the blood. Again, heavily regulated. All right, so the ependymal cells basically are using ion pumps to say, yep, you can come through, nope, you can't come through. All right, and then the cilia of the ependymal cells is helping to keep that cerebral spinal fluid moving as it passes through the ventricles. And again, if I come back to this image here, you'll see that the lateral ventricle is connected to the third ventricle, right, by this opening called the interventricular foramen. And the third ventricle is connected to the fourth ventricle by what we call this cerebral aqueduct. Right? And so all of this is interconnected to one another. And as I mentioned, we've got this thing called a blood-brain barrier, and the ependymal cells help to form this blood-brain barrier, as do the astrocytes. As do the astrocytes. Again, go back to video 1 of our look at the nervous system. The blood-brain barrier, and this isn't the only place we have a brain barrier. I will mention there's a thymus brain, I'm sorry, there's a blood thymus barrier, there's a blood uterine barrier, there's a blood testis barrier, and we have a blood brain barrier. Wherever you've got sensitive, highly metabolic, highly regulatory tissue, we tend to keep the blood separate. The blood is a little dirty. It's not as sterile as we would like it. There's a lot of antigens and antibodies and other things that are floating around in the blood that can be highly sensitive and triggering or damage to tissue. Um, and so we see this as a common mechanism. Right? Again, being highly selective for what can cross over into that neural tissue. All right? And so you can see here, the astrocytes are the primary cell type. The ependymal cells do aid this a little bit, but the astrocytes are the primary cell type that's creating the blood-brain barrier. It allows water, oxygen, carbon dioxide, alcohol crosses it very easily, glucose, yay, caffeine, nicotine, most of your amino acids go through with no problem. And they will cross that blood-brain barrier usually through simple diffusion or through what we call transcellular transport channels. And this is common. Right? This is common. Not everything gets across that. Testosterone does not cross that. This is particularly important when we look at things like fetal development. Right? And we talk about the masculinization of regions of the brain. Right? And sex identity that develops because of that during fetal development. And so not everything is allowed to cross the blood-brain barrier. Very often chemotherapies do not cross the blood-brain barrier. Ironically, we have what we call phage or bacteriophage that live within the brain that can actually numb, if you would, the blood-brain barrier and actually bring the blood-brain barrier down. And we've been able to harness that concept in treating brain cancer. And we can actually now create drugs that specifically, again, numb or bring down that blood-brain barrier to allow chemotherapy to get into the tumors to treat brain cancer. And then when the numbing wears off, the blood-brain barrier comes back again and all is well. This here is the difference between, again, your typical capillary and capillaries that are entering into that neural tissue and again, the formation of that blood-brain barrier. You can see those astrocytes, those podocytes, the feet of the astrocytes are just covering, completely protecting the capillaries within the brain. And again, this makes it very safe, if you would, or highly selective in what is leaving out of those capillaries and what is not leaving out of those capillaries. And so that is our look at brain and their associated structures. Again, a lot of this complements what we've already talked about in the lab. The last two images that I'm going to leave you with, and I was hoping to try to label them, but I don't know what's up with my keypad today. I'm not even going to try that, but you've got these two scans for you to go through and practice labeling. All right, what are some of the things that you want to label in here and be able to identify? Well, you can see the white matter. You can see the corpus callosum. You can see the gray matter. You can actually see the cerebral nuclei, right? These associated additional areas of gray matter. These are the lateral ventricles right here. Again, remember there's one in the left hemisphere, one in the right hemisphere. This is the third ventricle right here. So you can see these kind of things. Right? This is the cerebellum. Right? You can see the white matter, or the arbor vitae of the cerebellum. You can see the folia of the cerebellum right there. All right. You can see the subarachnoid space. That's this hollowed space right here. And so you can see and identify various structures. You can see the longitudinal fissure here. Right? You can see the gyri and the sulci, right, associated with that. This is of the the same scan lateral view. This was a former high school student of mine back in the day, previous life in New Jersey when I taught high school. She was a field hockey player, got bounced around a little bit, went for an MRI of the brain to see if she had a concussion. She didn't. She didn't believe the doctor. She brought the scans into me, and she's like, "Young, can you give me a second opinion? I'm not a medical doctor, but I was able to at least be able to sit there and say, yeah, no, there's yeah, good. She has a brain. I often wondered that at times throughout the school year, but there was no concussion. If there was a concussion, you would see compression up here within the subarachnoid space because the inflammation would happen within there, and that would press down onto the brain, and so you'd get this wider gap within the subarachnoid space. But again, you can see gray matter, you can see white matter, you can see the corpus callosum. Here you can see the thalamus, you can see the pons, you can see the medulla oblongata, you can see this is the spinal cord here. The pituitary gland is sitting right here. Right, so that means this is the just completely left. But this here is the pituitary gland. This is the infundibulum coming right down here, right, that you can see. Kind of cool. Here's the fourth ventricle over here. All right, third ventricle is over here. Lateral ventricles are going to be up over into this area here, up there. And again, you can see the cerebellum there as well. So this is a really good image and view of exactly what the brain looks like, and you can identify various structures on there. And so with that said, I'm going to allow you guys to continue doing your review and checking things out, asking questions as you see fit. Again, you can go back and review these questions, these review questions as many times as you want. And you know what I'm going to say? I'm going to let you guys say it this time. On a count of three, you can all say it. 1, 2, 3.